Hello, welcoming back the folks who are joining us on Zoom. If you can hear my voice, please make your way back to your computers. We're going to get started with our first agriculture panel of the day, which our Social Research Director, Tatiana Schreiber, will be moderating. So I'll pass it off to Tatiana for that.
Thank you. So just very excited to moderate this panel because I'm very interested in how, the practical applications, how urine is actually being used right now. Thank you, Julia. So we have three panelists for this first session on the urine use in agriculture. And so we only have three. We have a little bit more extra time, but I'm going to ask people to still try to stick to their 10 minutes more or less. So we have plenty of time for questions and answers. You're still having trouble hearing me? Okay, I will try to speak louder. So we start with Lucinda Lee. Lucinda is currently pursuing a PhD in environmental health engineering after completing her Bachelor of Science and then Master of Science in environmental health engineering. And she is going to talk to us about the application of urine-derived fertilizers for ecological nutrient management. And she can explain to us what that is. So go ahead, Lucinda.
Okay. Hi, everyone. Can you hear me? And
yes.
Everything's good? Okay. Great. Thanks, Tatiana. Yeah, so I'm really excited to share our work with ecological nutrient management of urine-derived fertilizers. This is work that I did at Michigan, where I have since completed my PhD, but I'm excited to share this work. And to start, I'd like to acknowledge those that supported this work, including NSF, the University of Michigan Graham Sustainability Institute, undergraduate research assistants, facility staff, and my colleagues in my research groups. So as we've talked about today, and most of us know, one of the strongest motivators for recovering nutrients from urine is the nutrient inefficiencies in our current food, agriculture, and waste systems. So here we see that a lot of the nutrients are brought into our food and agricultural systems, and most of it passes through. And that causes negative environmental impacts like algal blooms, and is why a lot of us are interested in this work. But here we can also see that it's estimated as much as 50% of the applied nutrients are lost from the field, making it the largest source of nutrient loss in these systems. And so while our efforts in recovering nutrients from urine and using it as a fertilizer helps us improve overall nutrient efficiency, we need to address the nutrient loss from agriculture. And overall in agriculture, there's a push towards using less synthetic or inorganic fertilizers to minimize nutrient losses to the environment. But there's by doing this, we also reduce reliance on inorganic fertilizers. And this can be done through an ecological nutrient management framework, which is an integrated approach that applies ecological knowledge to optimize soil fertilizer, crop production, ecosystem services, and long-term sustainability. So as we plan to use urine-derived fertilizers, we need to think about how they affect nutrient cycling as a means to prevent losses within this ecological nutrient management framework. The UDFs are typically classified as waste-derived fertilizers, just like biosolids and compost. But not only are they different in composition, the perception of them are quite different. So from this study, the surveys were done with about 2,000 participants, and they found that UDFs were perceived better than inorganic fertilizers, but not as well as organic fertilizers. But when actually used in studies, UDFs perform similarly to inorganic fertilizers because they're very similar in chemical composition. They have very high levels of soluble nitrogen and phosphorus that are easily accessible by plants. And that also results in similar nutrient use efficiency, which is the ratio of nutrient uptake by the plant versus the nutrients that were applied. And as we've discussed today in the past couple of days, there's an interest in pushing for UDFs to be approved for use in organic agriculture. So it's important to understand how UDFs fall on this spectrum of inorganic versus organic fertilizers. And because using UDFs are pretty similar to inorganic fertilizers, using them doesn't address this problem of nutrient loss from agriculture. So if you recall, about 50% of the nitrogen applied to a field never makes it into the food we eat. And so this provides an opportunity to improve the efficiency of fertilizer use so that we can consider UDFs in this ecological nutrient management framework. Previous studies have also seen that when you combine soluble fertilizers with organic fertilizers with high carbon content, for example, we can see an improved plant yield as well as soil health. And so this is what we're interested in doing, is how we can use UDFs with organic fertilizers in order to improve nutrient use efficiency. And for this research, we're mostly really interested in nitrogen as opposed to phosphorus because UDFs have a higher potential to offset nitrogen fertilizer. And so with that, our two research questions were, how do the impacts of UDFs on soil nitrogen cycling differ from those of organic fertilizers? And how can combined use of UDFs with organic fertilizer improve soil nitrogen cycling? So here we measured indicators of soil nitrogen cycling to better understand nutrient use efficiency. So in our experiments, we conducted greenhouse experiments to compare 6 fertilizer treatments. Each of them was applied with the same amount of nitrogen, except for the control. And so we have our control where there was no fertilizer. We have inorganic fertilizer, our urine-derived fertilizer, which we purchased from Rich Earth. So this is pasteurized and freeze concentrated. And then we also, in the lab, we also passed it through activated carbon to remove any organic contaminants. Our organic fertilizer, we chose composted poultry litter, which was available for purchase. And then our two treatments where we combined our soluble fertilizer, so that's the inorganic with the organic, and then the UDF with the organic. And with these treatments, we grew mustard plants for over 2 months. We applied fertilizers at the beginning of the experiments. And then throughout the period, we took samples for nitrous oxide emissions, which is a potent greenhouse gas, and can also signal the nitrogen activity that's happening in the soil, as well as leachate. So any of the water that passes through the pot, we collected. And so that's what that looked like, our setup. At the end of the experiments, we sampled the plant tissue and the soil to determine the nitrogen content in them, as well as measured nitrogen functional genes that represents different steps along the nitrogen cycle. So that can be seen here. And so from our experiments, we saw that overall plant biomass and plant nitrogen were the same for UDF and inorganic. The compost treatment had the highest plant biomass and amount of nitrogen in the plant tissue, while adding compost to inorganic and UDF increased the plant biomass and nitrogen mass. They were not significant. And so you can look at the letter that's above each bar, as that represents significance groups. And we were surprised by these results because a lot of other studies report that organic fertilizers in the short term did not increase yields and sometimes result in lower nitrogen uptake by the plant. And we think that our results might have been caused by the type of organic fertilizer. We use a poultry litter compost, which tends to be higher in nitrogen content than other organic fertilizers. For our nitrogen losses, we found that the nitrogen that's lost as leachate was not different among the treatments, although it was slightly higher. If you're looking at just the average, the highest in the UDF treatment. When we looked at losses as nitrous oxide emissions, we saw that the compost had the highest emissions. Also important to note that the inorganic fertilizer and the UDF had similar nitrous oxide emissions, but there was a significant increase when compost was added to both of those treatments. So you see that increase here. From our qPCR results, where we were measuring nitrogen functional genes, we saw that there was a significant increase in these different genes that represent different steps of the nitrogen cycle between the control and the compost, which signals to us that the increased nitrifier and denitrifier abundance in the soil could have been caused by the organic carbon additions from the compost, and that would translate into higher nitrous oxide emissions that we observed. Finally, when we compare total nitrogen loss as nitrous oxide and leachate combined, we found that the highest loss, again, was for the UDFs alone, but this was not statistically significant. And on the right here, if you take a look at the ratio of nitrogen loss over the nitrogen uptake by the plant, we found that that ratio was significantly lower when UDF was combined with compost as compared to UDF alone. So this means that there's less nitrogen loss for every unit of nitrogen taken up by the plant for the UDF with compost treatment as compared to the UDF alone. However, there can be a global warming potential trade-off. And so I'm popping up the nitrous oxide emission data up here again. Nitrous oxide emission, like I said, nitrous oxide is a potent greenhouse gas. And here we see that when we added compost to both the inorganic and the UDF treatment, we get this significant increase in nitrous oxide emissions. So it's something we need to consider in terms of overall environmental impacts if UDFs were used with compost. So in conclusion, we found that the UDF we use perform as well as the inorganic fertilizer, and they're more similar to each other than UDF is to compost. We also found that compost outperformed in yield, but also led to higher nitrous oxide emissions. And finally, combining UDF with compost reduced nitrogen loss to nitrogen uptake ratio. And there's a lot of future work that can be done here, including understanding the nitrogen losses from different types of UDFs, of all the different ways we can process urine that we've already seen today. We can also think about evaluating different types and combinations of organic amendments with UDFs. And currently we are writing up the work, and we can share that study with everyone when it is published. And with that, I'd like to thank everyone for your attention and for Rich Earth for hosting.
Thank you, Lucinda. Okay, and now we have, next panelist is Rebecca Nelson. And Rebecca is a professor at Cornell University College of Agriculture and Life Sciences. Her interests focus on sustainable agriculture and food systems with research activities and engagement in Africa, India, and New York. And her research group has 4 main focus areas: the circular bionutrient economy, mycotoxin management, disease resistance in crops, particularly maize, and agroecology and food systems. And her talk is on Grow As You Go, container-based sanitation meets container-based gardening. So she'll be talking about her Grow As You Go gardening project. So go ahead, Rebecca.
Thank you so much. Appreciate that introduction. And I really want to thank the Rich Earth Institute for hosting this wonderful event. I think it's become a really important part of the annual cycle for many of us interested in this topic. So thanks, Tatiana. Thanks, Rich Earth. Thanks, everybody, for being here. So yeah, the basic proposition that I want to put forward is this idea that we can combine low-value carbonaceous materials, in particular cereal residues, with urine to create high-value organic matter that can be used to produce food sort of simultaneously, that carbon plus NPK going to compost, going to food. And so I thank Lucinda for sort of setting that up, and also John Culpepper's proposition of yesterday being very relevant. So the context for this proposition is sketched out here. We have about 40% of the world's population living in peri-urban areas, frequently without access to services such as sanitation or good food security or employment opportunities. People in the peri-urban areas are sort of proximal to rural waste, such as crop residues, or rural byproducts, such as crop residues produced in low population density areas, and the excreta produced by the higher density urban areas. So can these people who are often producing food in contaminated soil conditions or very crowded conditions benefit from those true, perhaps surplus products on either side of them? So inspired by this brave need and effort to produce food in bleak peri-urban areas, as illustrated in this photo from Kenya, the following questions have kind of arise. Can we use urine as a fertilizer for this on soil or on soil residues? How does that dynamic change as the material changes over time? Indeed, what sort of substrate-based recipe does it take to get good, effective food production? What's the format that's suitable for these contexts? And how do these options interact with the different contexts that we might experience in the United States and Kenya or anywhere? I fiddled around with straw bale gardens for a couple of years, just seeing how it went. And then last summer, we started getting more into serious experimentation. So I'll present some findings from our high tunnel experiment here on the Cornell student farm, and then also from a few ancillary experiments, this Sacks on Racks experiment next to the high tunnel, and from a few little experiments elsewhere in the US and also in Kenya. So first off, comparing P and conventional fertilizer, we did not see a significant difference in terms of plant performance on the different matrices, with one exception, which I'll see in the next slide. So P and conventional fertilizer were different. I mean, we're the same, which is great on either soil or maize stover. Looking at the kale dry weights, and shown here, the kale dry weight for these three substrates, we see that the soil beat the maize stover, which beat the wheat straw, particularly when the wheat straw was fertilized with urines, which is really pathetic. So the first season of wheat straw was really, really bad, but stover was better. Then we did nutrient analysis for both the kale and for the substrates, and none of the chemistry really gave us the same explanatory sort of behavior that we're seeing in terms of yield. The kale had the lowest tissue yield on soil fertilizer fertilized with P, weirdly enough. So it didn't seem like, that's a very high yielding one with low nutrient availability, nitrogen in the tissue. It looks like the nitrogen wasn't limiting the plant growth, though. Eli Newell, undergrad, had the idea to look at the salt concentration that might build up. And indeed, we do see higher levels of salt in the kale tissue when treated on wheat straw based over in soil when treated with urine, actually, but not significantly saltier kale on soil. And similar patterns to what you see here are seen when we analyze the sodium in the substrates themselves. When we looked at the substrates that were out of the high tunnel on that Sacks on Racks experiment, where there was a lot of rainfall, we did not see that buildup of salt. Looking at phosphorus, we see that the maize stover has higher level of phosphorus than the wheat straw, and that, again, we're seeing lower phosphorus on the soil than on the Miracle when treated with P relative to Miracle Grow. Calcium, a similar weird pattern where you get this trend. They're all not significantly different, but there's a trend for higher calcium with a P, but much lower calcium when soil is treated with P. So the behavior on the organic substrates was quite different from on the soil. And the same pattern was actually seen in the matrix, the substrate, as well as with the tissues. So then stepping outside the high tunnel and looking at the experiment on the Sacks on Racks, we're looking here now at the behavior of, this is a lettuce experiment on a series of substrates that are sort of differing in age. So virgin wheat against weathered wheat, where the same wheat bales were in the barn or out in the elements for a year, composted in the pea straw bale garden for a year, and then comparing that with freshly ground maize stover and a mix of the composted wheat. And you can see that the wheat goes from being pretty terrible to medium to on par with freshly ground maize stover once it's been composted with urine. So there's different requirements for this conditioning process to get a decent substrate for growth. But the Les was quite happy on any of these more mature materials. And if you compare the char versus no char, the two different colors of green, we did not see a significant biochar effect in the high tunnel experiment or in this out of the high tunnel experiment, presumably because there was no water or nutrient stress applied to these experiments. Now moving over to Kenya, we're working with Charles Midega over the summer. We had a team there with Eli Newell and Wendy Alwala, Cornell undergrads. And here's a sack experiment with kale comparing urine and water. And we see significantly better performance when the sacks are fertilized with urine than with water. But we do see a significant effect of the highest, the 10% biochar did give us superior kale growth there. Coming back to Ithaca campus, looking at an okra experiment here, looking at ground maize stover with and without pea and biochar. When we had both pea and biochar, we'd get good plant growth. If we just had pea without the char, in this particular experiment, we managed to kill all the okra plants, presumably by sort of overdosing the substrate initially. And then we did, without the pea or biochar, we'd get very pathetic little plants growing on the substrate alone. This is, I was working together with a visiting scientist from, a visiting entrepreneur from Burundi to do these experiments, Jean-Marie Nizigimana. So then the other experiment that Jean-Marie and we did was comparing rudely chopped versus finely chopped ground maize stover. And I was so convinced that this was going to be very important that we put a lot of time, energy, and money into getting a nice grinder over for the Kenyan operation, only to find that there was absolutely no difference. Like the maize stover, whether chopped rudely or ground, chopped straight by, during the harvest process or machete type job, was quite a good substrate just because it composted with the urine so rapidly. So really didn't need to have made that considerable investment. So the last data slide here is looking at, I was sort of frustrated. We don't really have a best guess substrate. Our best guess in the sort of late in the season was 80% compost wheat, 20% food compost, and it marginally outperformed commercial potting mix when everything was fertilized with urine. No significant char effect. We did try 0.5 and 10% char, no effect. Again, no stress, no effect. And just to say, this is about 120 liters of material per, say 100 liters of material per sack. At $1 a liter on Amazon for these sacks of organic media, man, I think that being able to do it yourself with these low value organic underutilized resources, our stuff, could really be a benefit to people with large volume propositions or low resource propositions. So thus far, the take-home message would be pea works well as a fertilizer. Straw needs more conditioning with pea than conventional fertilizer requires. We did not need to grind. We could just rudely chop our maize over and do okay. Mixing composted wheat and compost did as well as the commercial potting mix with pea fertilizer, gave us good tomato crop. And biochar helps, but only if you have stress to help with. So then next question that had come up was like, what about formats? So I fiddled around at our off-campus art science venue, the Soil Factory, and we got sacks on racks, sacks made out of trousers, bins of various kinds, spools on pools, pools on spools, excuse me, all kinds of zany situations. But really it was these bales as rails that sort of ended up in the season seeming the most reputable. I'd done this the previous year, did that, the rhubarb and herbs were this year, that's potatoes last year, and setting up a new bale array in 10 minutes at the student organic farm. So the idea is you frame up a raised bed, costs 1/10 the price it does to build a raised bed with wood sides at the organic student farm. So you just can quickly and participatorily put up a raised bed, fill it with your compost and last year's bales. So what I did was you've conditioned the frame with urine throughout the season. So you do have that aging composting process going, then peel off the peel off the frame and reframe the bed with fresh bales each year and use the old bales as a growth medium for your next crop. So we're quite excited about our new straw bale array at the soil factory shown here. We're in the process of filling this up with our compost and leaf waste for the year, and we'll be dumping pea on that through the winter or at least in the spring fall and spring to compost the inside, and then during the growing season to compost the outside. And I'll keep you posted. So the idea is, could this be something that could be useful to turn a vacant lot in a city rapidly and through community participation into a nice pea-powered garden? So then quickly shifting over to the East African context, we have the Circular Bionutrient Economy Network. So Johannes Lehmann and Bella Culotta have been coordinating and getting us going for this network. Here we see our collaborator, Charles Midega, talking to a representative from the local city's sanitary facility, the Benjamin Odhiambo, and a representative from Sanergy Fresh Life here, admiring that Benjamin's toilet, which has black soldier flies eating the material in the solid chamber, and then the pea comes out and is dried in this little greenhouse thing. Here's an economist from the University of Nairobi doing a experiment in Kibera, one of the large, and this is Bella Culotta working with Wajuku to make a pea-powered garden in another informal settlement, 2 informal settlements of Nairobi here shown. So after that, so that was in January last year. We'll have our next CBEN, Circular Bionutrient Economy Network meeting in January. Come one, come all, or at least register. We'd love to have you there, and meeting in Kisumu to advance the East African cause. But in the summertime, I find myself back at an organic agricultural training center in Kitale, the Manor House Ag Center, and I'm talking with one of the people that work there, and we can't see our entry point. We've been working with them for a couple of years. Nothing's really taking off. He thinks there's too much taboo, so we should just go with the arborloo, plant a tree after you move the outhouse. I'm pushing for using pea as a fertilizer on plants, or hot quenching biochar produced in a Kontiki oven in urine to get a hot charged fertilizer. But then none of them seem to be taking off. No one seems comfortable with any of those options. So we go around and talk to a lot of farmers, and what they're excited about is power composting. They're using something they're calling bokashi compost, chop fine, add expensive ingredients, and toss it twice a day. Looks like a pain in the neck, but they're willing to do it with the high cost of fertilizer. So we started working on this idea of pea-powered composting more seriously. Patrick and us, we set up some experiments at Manor House, and he was so delighted with the results that he started working really seriously with farmers to use that as a fertilizer, given the high cost of conventional fertilizer. And the results were so good that farmers started directly applying urine as well as making pea-powered compost. So that seemed to be a good entry point for us, the pea-powered compost. Coming back to Ithaca, I kind of got into the idea of, well, we build compost piles and toss them around too much, and green plus brown, let's go brown plus yellow. So started talking with my extension colleagues about that, and people seem interested. So we're interested in fancy potties. We're interested in the idea of a pea pod collecting. We did our first festival collection. Here's see the rich earth propaganda here and different academic studies. I reeled in quite a bit of pea during this event. Plus, I reeled in these 2 engineering students who are working on microcontroller now with Lucinda to improve our various experiments using microcontroller technology. So just starting to wrap up here, where our main outreach work really happens at the soil factory site, where we have art, science, community stuff. There's some art kitty fun projects that we've done. Here's us at a recent event with Rich Earth participation, talking to our local legislator, our Anna Kellis, our New York State Assemblywoman, about we're trying to figure out how to get our biosolids to biochar. This is a collaboration with Rich Earth, and so we can avoid PFAS, avoid wasting nutrients. So just sort of coming back to our questions with a few answers, I think we have gotten some ideas about the recipe. We see that if we let different substrate need to mature for different periods of time, but they become good substrates over time, especially when mixed with compost. We'll have to get with Lucinda about the greenhouse gas implications. Urine is a good fertilizer, as I think everybody on the call knows, but we do see there's some potential deficits and some salt issues that might need consideration. Explorations of SACs have led me to this bales as rails and trying to figure, how to make bundles of corn stalks to make it work in Kenya. And, how do we work with people, gardeners and farmers and here and Kenya and around the world? Really excited to go forward. Thanks so much to the team, to our funders.
Thank you, Rebecca. That's great. All very interesting.
So I'm afraid that I have class now, so I'm going to have to dash off. And so I'm hoping that my colleagues, Lucinda, Christina, Bella can handle any questions that arise, because I gotta be in front of the class.
Or we'll send you some questions later and you can respond to them. But thank you very much.
Okay, thanks so much.
Okay. Our next presenter is Fernando Perez, and he has a PhD in technologies for waste treatment and reuse at the Swedish University of Agricultural Sciences. He's a researcher at the University of San Simón in Bolivia, and he's collaborating with a group of environmental engineering group from SLU in developing the drying technology for treatment and reuse of urine. And his talk is entitled Simulating Reverse Blending of Dehydrated Human Urine Fertilizers with Organic Waste to Meet Macronutrient Demand of 15 Major Food Crops. So go ahead, Fernando.
Hi, can you hear me?
Yes.
Okay, thanks. And I hope you can see my presentation now.
Yeah, it looks great. Thank you.
Okay. Good morning, good afternoon, good evening, wherever you are. And thank you for inviting me to share our findings in this topic. So I'd like to start by saying that our overall goal here is using urine as a fertilizer, right? And at the end is substituting the synthetic fertilizers with the recirculated nutrients. Now it has been shown globally that 25% of the Haber-Bosch nitrogen and the 25% of the phosphorus that is mined from rocks could be replaced by urine, right? Now, within this framework, dehydrating alkalized urine, which is the technology that is being developed by SLU, produces a powder that has these concentrations, like around 18% nitrogen, 1.6% phosphorus, and 4.6% of potassium. This is comparable to some commercial synthetic fertilizers. Now, in several publications, it's been mentioned that the balance of nutrients in urine is similar to the balance required by crops. However, the crops have a wide variety of NPK requirements, as we can see here. We see here the ratio of the three macronutrients in the dehydrated urine, which is the green, and the ratios required by 12 crops that you can see here. Now, due to these differences, if we would try to fertilize crops only with dehydrated urine, the results would be nutrient supply in excess, which has, as we know, significant environmental consequences, not just because of the nitrogen excess, but because of the phosphorus excess. Now, depending on the crop, the excess of the nutrient supply via dried urine can even double the crop demand. So dried urine must be combined with other renewable fertilizers to achieve optimal yields, the crops, and also environmental sustainability. So the purposes of our study were to know what organic wastes can be blended with dried urine, what are the application rates of these blends, and if we can control the excessive nutrient application by using these selected blends. So to achieve that, we simulated the nutrient contents in blends made out of dehydrated urine and anorganic waste, one organic waste, by using an approach known as reverse blending. So we performed we basically performed a literature review looking for organic wastes with NPK concentrations reported. We then simulated the NPK ratio in blends and selected those whose ratio was similar to that required by 15 measured crops. And then we calculated the mass of the selected blends to be applied to each crop. So about the first question, we can see here the list of 38 blends that would match the needs from 15 major crops, including the organic waste and its corresponding percentage with blending it with dehydrated urine. So there's a lot that can be said about this, but the key message for this presentation is that out of these 38 selected blends, 20 are formed by ash, would be formed by some type of ash, and 10 would be formed by some biochar. In other words, 13 out of the 15 target crops could be fertilized with blends of dehydrated urine and either ash or biochar. Now, the main characteristic of both types of materials, I mean, ash and biochar, is the low contents of nitrogen. In the case of ash, it's normally up to 0, and for biochar is generally less than 3%. In dried urine, the ratio of nitrogen to phosphorus and potassium is high in dehydrated urine. So blending dehydrated urine with ash or biochar results in blends with lower ratio nitrogen, phosphorus, and potassium. I mean, these proportions are lower, obviously, than pure dehydrated urine and would match the crop demands. As regards to the remaining questions, the answers actually are crop dependent. So we cannot examine here the 15 crops, so let's examine a couple of them to see the results and to draw some patterns from them. So we see here the macronutrient proportions for the 3 selected blends. I know that it's small, but these are the 3 selected blends. And for a synthetic fertilizer, which is the small diamond, purple diamond, and the dehydrated urine, which is this triangle. All are compared with the demand from barley, which is the black asterisk. So the mass to be applied is displayed both here in the color scale and also in the numbers that are here in the legend. So what we can see first is that none of the macronutrient proportions, none, I mean, neither the blends nor the synthetic fertilizer would perfectly match the barley demand. However, the application rates blends are about 50% higher. 50% higher than synthetic fertilizer, which is caused by the higher concentration of macronutrients in the synthetic fertilizers, of course. And about the nutrient excess, with the blends, the nutrient excess of nitrogen and phosphorus are reduced to almost 0, while there is some potassium excess. For the synthetic fertilizer, there is some potassium and phosphorus excess. For maize grain, the application rates of the blends are similar, as we can see here, and even lower than the synthetic fertilizer. In this case, the synthetic fertilizer at the required application rate would produce an excess of nitrogen and potassium. This is explained by the reverse blending approach, which we use, and this approach allowed us to suit the blend formulations to the crop demands, which is not the case for pre-formulated synthetic fertilizers. Now, in general, we found that it is possible to control the excessive application of nutrients with the blends for several crops. The main limitation in this case would be finding an organic waste whose nutrient concentration combined with dehydrated urine matches the crop demands. So as conclusions and answering each question, what organic waste can be blended with dehydrated urine to meet the NPK requirements? It's basically ash. We found that ash and biochar are suitable for most crops because of the low nitrogen content. And what are the application rates of the blends? these are variable, but in general, are higher in 50% than the application rates with synthetic fertilizers. But we also found that it's possible to have blends with application rates similar to synthetic fertilizers. Now, if it's possible to control the excessive nutrient application, we concluded that it is. In many cases, the excess with blends were found to be lower than with synthetic fertilizers. This depends on the crop demand and the organic waste availability and data availability. Finally, a sort of recommendations for future work, of course, more research on nutrient content in organic wastes and on the blending dehydrated urine with 2 or more wastes is needed, because we found in some preliminary results where the mass of blend that should be applied is way lower when we use 2 or more wastes combined with dehydrated urine. Because this would reduce, hopefully, the application rates and nutrients. Yeah, and that was a presentation. Thank you, and here's some information to contact us.
Thanks so much. That was great. So those all fit together very nicely and complemented each other. So I think that was very, very interesting panel. We have time for a few questions. So I don't know if we want to start with a question from the room. Yeah, so there's a question for Lucinda, which is similar what a question I was going to ask. How you, the question was if you applied the exact same nitrogen rate in all of the experiments, so in all of the treatments that you use, so knowing, as you mentioned, that poultry litter is very high nitrogen, did you balance that out in terms of having the same nitrogen level for each treatment?
Yeah, so we applied the same rate of plant available nitrogen. So that would be the sum of ammonium, nitrate, and mineralizable organic nitrogen.
So that included the nitrogen content of the poultry litter? Or is that,
Yes.
okay, I see. Okay.
Yeah, so I measured the nitrogen content of all the fertilizers in the different pools, and then I calculated the plant available nitrogen of each of them and then adjusted accordingly.
I see. Okay. Great. That was very interesting. Arthur has a question. Go ahead.
Yeah, my question is also for Lucinda. Just going back to the beginning of your presentation when you were talking about nutrients wasted from agricultural use. And I was wondering, with just thinking about, I feel like I was wondering how much of that, if you have any data on how much of that is based on like overland runoff versus leaching, because obviously with a lot of the uses of urine-derived fertilizer, there's really no overground runoff because it's a liquid. If that question makes sense.
Yeah, and I think this is kind of similar to, or at least related to a question that Abe asked in the chat, which is just the different nitrogen loss pathways when you apply nitrogen fertilizers, particularly. So some of that gets converted to nitrous oxide emissions. Some of it is lost as runoff or leachate. I don't have the numbers, but I think there are studies that have done not full mass balances, but at least estimates of where all of that nitrogen goes. I think, for example, Jennifer Blesch, who was involved in our work, has a study on that, as I'm sure there are others too. But yeah, that's a good question.
Okay, I think we had another one from the Zoom. Okay, one more from the room.
Hi, my question's for Fernando, and I first want to comment that it's really useful, the display of the triangles for the NPK and comparing the needs, the demands of the plants and the different fertilizers, and I'm curious if that's going to be available, those images for reuse, or if you've published them yet, what that plan is. And then my other question for you is, moving forward, are you looking to implement this in a field study / in a community, and what might that look like on the ground?
Yes. Thank you. Yes, actually, we are. This is this is just a small part of a paper that we are preparing. That, of course, we hope that it's published. And yeah, actually the database, because and this is also to reply to some question in the chat. In looking for these materials, we prepared like a database with around 400 different materials with NPK concentrations. And yeah, all of this material is going to be available as supplementary information probably in that paper when we publish it. So that was one thing. The second one is, yeah, we are using the results as a sort of guide. I mean, we are part of this team, SLU at Sweden. They are actually in field trials as far as I know, but I am not sure if they are trying with one or two crops. I mean, someone from SLU here can say that, but the idea was to have a guideline to what happens with other crops in other parts, to have an orientation to look for some patterns. So I hope that I mean, I know that there are some projects running in other parts of the world with alkalized dried urine. So I hope this is going to be a guideline for future implementations.
Great, thank you.
All right, well, we're about at time, but there are a lot of questions that were answered in the chat. So it looks like it was a lively discussion. I can't really see it from up here, so I couldn't really see, but I got a bit of a sense of that. But thank you for all the panelists. And now we will break for lunch, and then we'll come back and continue with talking about applications in agriculture.
Great. Thank you, Tatiana. Yeah, so we'll I'll reconvene back here at around 1 p.m. So if folks on Zoom want to take a break and come on back here, then we'll have the second part of our agriculture panel start off at 1. So look forward to seeing you all back here then. Thank you so much.